Hello, do you have my voice and video? Hello, do you have my voice? Yes, okay, thank you. And can I upload my slides?
Say again.
My slides. Okay, bye. Thank you. Bye for now. I'll try it now. Where should I upload it?
First, open your slide. Kızım bir ekran paylaşmayı bilmiyormuş da onu deniyorum. Bir Zehra. Zehra, can you hear me? Yeah. You turn on your voice and
Okay.
now please try to change the page.
To change the page. Okay, yes. 1 moment. This is the slideshow.
But we are just seeing your first page now.
And
Just
you see the slides, yes?
I see your first page. Sometimes it is stuck just during your slide. We are just seeing first page. That's why try to change it.
Okay, I'm changing.
But we cannot see it.
The other slides?
Just we are seeing your topic page.
Just topic. Okay. So what should I do?
Exit from full page, full screen.
Okay.
Manually change. Try it again.
Okay. You know, my Zoom has covered the all.
Just try. I am waiting. Don't worry.
I'm
Try during
sorry.
the presentation. There won't be any problem. Just open your presentation and then screen share. Screen share button.
It's going to be better. One moment. I'm going to reshare my slides. Okay. Okay. Now you see my slides?
No.
No.
No.
And now?
Not yet. Are you clicking to green screen share button
Screen share.
on the Zoom?
You know, I don't see the
Down,
screen share
down,
button.
place, down, place.
Down. Hmm.
Green.
Yes, share screen. Okay. Now
Now
you
I
see my
see, but
slides?
don't make full screen. Just manually click.
Okay.
Manually change.
Is
Yeah,
it okay?
it is okay now. Perfect.
You see. Okay. Thank you.
See
Thank
you.
you very much.
Yeah. See you during your presentation. We will start at 3:30. Half past.
3:30.
Yeah.
Oh, thank
Okay.
you, Damla.
Bye bye. Good luck.
Good luck. Thank you.
Şimdi ses geliyor.
Geliyor mu ses şu anda? Ha, sesini açmışsın sen gel. Tabii geliyor.
Dear respected researchers, welcome to our second session of the today. I would like to thank all of the researchers and academicians here. Now our moderator is Associate Professor Dr. Bekir Oğuz from Van Yüzyıl University. I would like to give the speech to him and I would like to thank to him for his help for the Congress. Yes, Professor, your turn.
Evet, herkese merhabalar diyorum. Bugün sanırım oturumumuzun, bugünkü kongremizin son oturumunda beraberiz. Sizlerle beraber olacağım bu sunum boyunca. Çok fazla uzatmadan isterseniz ilk sunumla başlayalım. Değil mi Damla Hocam? Sıkıntı yok.
Evet hocam.
Tamamdır.
Uygundur.
Tayfun Hocam buradaysa sunumunu hazırlayıp başlayabilir. Tayfun Dikmen Hocam.
Merhabalar herkese.
Merhabalar.
Ekranımı paylaşıyorum bir saniye.
Tamam, bekliyoruz Tayfun Hocam.
Hello everyone. I think you can see my screen.
We see everything's perfect. Good luck.
Okay, thank you. My presentation is amniotic fluid-derived stem cells as a promising stem cell type, isolation, characterization, and interpassage comparison. As you all know, stem cells are the cells that have capacity to self-renew and to differentiate into tissue-specific cells. And we can divide stem cells into 4 categories as totipotent, pluripotent, multipotent, and unipotent stem cells, depending upon their cellular potencies. Although totipotent and pluripotent stem cells has much higher cellular potencies, there are great concerns upon their derivation. There are great ethical concerns. And therefore, researchers are focusing on multipotent stem cells for stem cell research. Amniotic fluid-derived stem cells is one of these adult multipotent mesenchymal stem cells. And they have some certain advantages as they have less ethical concerns upon their derivation, and they're very easy to obtain. Also, they have relatively higher cellular potencies compared to other mesenchymal stem cell sources, as most of the researchers places them between pluripotent and multipotent stem cells in the context of cellular potency. In the study, we are going to talk about isolation and culture of these cells, and we are going to discuss some characterization studies, which are multilineage differentiation, growth curve analysis, immunofluorescence staining, and real-time PCR analysis. And after doing these, we are going to make a little interpassage comparison. For the isolation of these cells, we used Wistar rats, and at the 17th day of pregnancy, we collected the amniotic sacs in a sterile petri dish, and after washing them, we punctured these sacs in a place where we see no vascularization to avoid any blood contamination. After collecting all the fluids in a centrifuge tube, we centrifuged it at 250 G for 10 minutes, and we diluted this cell pellet with the culture medium, which was DMEM low glucose, supplemented with 20% FBS, 1% penicillin streptomycin, 0.1% amphotericin B, 5 nanograms per ml EFGF, and 1% L-glutamine. We cultured these cells in 37 Celsius degrees and with the supplementation of 5% carbon dioxide. At the initial passage, we saw a diverse morphology in the cells as we both see fibroblast-like cells, stromal cells, and epithelial-like cells. But right after the first passage, fibroblast-like morphology had become the dominant morphology, and they kept continued through the further passages. For multilineage differentiation, we conducted osteogenic, adipogenic, and chondrogenic differentiations. And after 21 days of induction, we confirmed these differentiations by their respective stainings. As you see here, we can see calcium deposits stained with Alcian red as staining to confirm osteogenic differentiation. Here we see oil droplets stained in red by oil red O for confirming autopogenic differentiation. And here we see bright blue glycosaminoglycans to confirm chondrogenic differentiation after Alcian blue staining. For growth curve, we compared the cells of passage 3 and passage 5 for 14 days. The cells of both passages had entered a logarithmic phase right after the 2nd day, and they both reached the peak at the 6th day of culture. And after this point, we see that both passages kept in a plateau phase through the end of the study. We see no differentiation in the context of proliferation pattern as well as the cell numbers here. For real-time PCR, we checked mesenchymal, hematopoietic, and pluripotent markers. For mesenchymal markers, we checked CD90, CD105, and CD44 expressions. Both of these genes were positive in PCR studies. And for CD105 and CD44, we see a stable expression. But for CD90, we see a significant upregulation at the passage 5 cells here. For hematopoietic markers, we checked CD34 and CD45. And despite we see a positive expression at the initial passage, these expressions were significantly downregulated in further passages. And for CD45, this downregulation further kept going at the P5 cells. We said that these cells had some pluripotent properties, and therefore we checked the gene availability of SOX2, OCT3, 4, and NANOC. For SOX2, right after the initial passage, we see a significant decrease in the expression. But in the context of OCT3, 4, and NANOC, we saw some significant upregulations at passage 5 cells. We also checked the protein availability of OCT3, 4, and SOX2 by immunofluorescence stainings at passage 3. And we saw that all the seeded cells were positive for these expressions. So what does these markers mean? SOX2 is an important enhancer of ectodermal differentiation capacity of cells, but also known to have some negative correlation with mesodermal differentiation capacity. OCT3/4 is one of the most important pluripotent markers, as we know that it has a fatal role in conservation of pluripotent state of embryonic cells, and it is also known to increase the mesodermal differentiation capacity of the cell. NANOC, in the other hand, is another important pluripotent marker, has negative correlation with hematopoiesis. So by the enlightenment of these outcomes, which passage should we use in our research? Of course, it depends on what you are looking for, but in the context of cell proliferation, we see no difference. And if you are in need of more cells, using these cells in later passages could be advantageous. But if you are looking for something like neurogenic differentiation or any other ectodermal differentiation, using amniotic fluid-derived stem cells in early passages could favor you, because we know that SOX2 is an important enhancer of this ectodermal differentiation, and the expression of SOX2 is being downregulated in further passages. But if you are looking for something like osteogenic or chondrogenic differentiation, which we know that they are mesodermal tissues, using amniotic fluid-derived stem cells in later passages could be more advantageous, because we are going to have more OCT3/4 expression and less SOX2 expression, which favors mesodermal differentiation. And also by doing this, we can avoid hematopoiesis potential, as we both see upregulating NANOC expressions and downregulating hematopoietic marker expressions. Thank you for listening to me. I had tried to give a brief information about my research. If you have further questions, I'm happy to hear them in questions section. Thank you.
Evet, hocamıza sunumu için teşekkür ediyoruz.
And I would like to warn everyone that you can type your mail address to the chat box, and if there is any question, you can write in the chat box, or later, if somebody reminds any question, they can directly contact with you. Thank you. Sayın
so
hocalarım,
much.
maillerinizi isteyenler chat kısmına da yazabilirler. Böylelikle soruları direkt size de yöneltebilirler. İsteyen buradan da yazabilir sorularını. Ama bazen daha sonra aklınıza takılan sorular olabiliyor. Onları da direkt araştırıcıya sorabiliriz. Çok teşekkürler. Buyurun Bekir hocam.
Evet. Hem de bu şekilde bize biraz daha hız katar chatten yazarlarsa. İkinci sunumumuza geçelim. Zehra Hesari. Burada kendisi.
Yes, I'm here. Hello.
Hello.
Should I start my presentation?
Okay.
Please.
Okay. Do you see my slides now?
Yes.
Okay. In the name of God, hello, and thanks to the organizers of the conference and all participants. I'm Dr. Zahra Hesari, Assistant Professor of Pharmaceutics in Gilan Faculty of Pharmacy from Iran. I'm going to talk about a magnetic platform consisting of spion-loaded PLGA nanofibers for neural differentiation of mesenchymal stem cells. As you all know, spinal cord injury is a complete or partial damage to the spinal cord, and the mortality risk is highest in the first year after injury. It costs heavy economical and moral cost for the patient himself and his family and society as well. Unfortunately, until now, there is no certain complete treatment for spinal cord injury patients except some drug therapy or medical devices, supportive cares. But tissue engineering and regenerative medicine nowadays has bright insights toward the treatment of the central nervous system injuries. One of the main principles of the tissue engineering are the scaffolds. The biocompatible scaffolds should possess particular biological, mechanical, physical, and chemical characteristics. A gold scaffold is the scaffold which can mimic the extracellular matrix structure of the target primary tissue, and it should provide the necessary microenvironment for the neural tissue repair. As well, magnetic nanoparticles have been one of the most attractive nanomaterials for various biochemical applications, including magnetic resonance imaging, diagnostic contrast enhancement, magnetic cell separation, and targeted drug delivery. They enhance mechanical and biochemical properties of the nanofibrous scaffolds, followed by an increase in cell proliferation. In our study, we used the electrospinning technique for fabrication of PLGA nanofibers. PLGA is a famous polymer for its FDA approval for use in human as an in vivo polymer that we fabricated 3 samples, the PLGA polymer containing 0% magnetic nanoparticles of iron oxide. Sample number 2 was the sample which contained 5% magnetic nanoparticles, and sample number 3 was the PLGA nanofibers containing 10% of magnetic nanoparticles. The scaffolds were evaluated with various analysis methods such as VSM to investigate the magnetization variation and magnetic response of the magnetic scaffolds, scanning electron microscopy to evaluate the morphology of nanofibrous structures and the average diameters of the nanofibers, and contact angle to evaluate the hydrophilicity of nanofibrous scaffolds with various concentrations of magnetic nanoparticles. Tensile test was performed to evaluate the mechanical property for the scaffolds with different percents of magnetic nanofibers. The stem cells were mesenchymal stem cells which were bought from registered cell bank in Iran, and they were passaged and used when they reached to a proper confluency. The scaffolds were placed in Petri dishes, and the cells were seeded on different types of scaffolds and evaluated for 8 days. The assessments included MTT assay for evaluation of biocompatibility of all 3 samples, all 3 scaffolds, quantitative real-time PCR with 3 neural markers, and immunocytochemistry staining with 2 neural markers as well. Results showed that according to magnetic characteristics, VSM results showed that the magnetic hysteresis curve of the spion PLGA nanofibers at room temperature presented magnetic PLGA scaffolds were considered to preserve the ferromagnetic behavior with a saturation magnetization of around 9. It shows that the scaffolds possess magnetic behavior without need to external magnetic fields. As you see the SEM images of the scaffolds in this slide, images A and B are related to PLGA scaffold with 0% of magnetic nanoparticles. Figures C and D are related to PLGA scaffold containing 5% magnetic nanoparticles, and images E and F are related to nanofibers containing 10% magnetic nanoparticles. Contact angle test of the scaffolds is shown in this slide, which shows that the plain PLGA scaffolds with no magnetic nanoparticle had the highest lipophilicity and the highest contact angle, while PLGA containing 5% magnetic nanoparticles showed a higher hydrophilicity in comparison with sample A. And when we had the PLGA plus 10% magnetic nanoparticles, we saw the highest hydrophilicity and the most appropriate scaffold as cell biocompatibility. In this slide, we see the tensile strength results, which shows a typical stress strain curves obtaining from the tensile test. The increased stress levels over the entire strain range is revealed for magnetic nanocomposite fibers. The results from the tensile test presents a ductile behavior for both magnetic and non-magnetic fibers. Although the inclusion of the spion nanoparticles strongly reduced strain percent, the results suggest that the modulus were increased. In addition, the elongation percentage of PLGA containing magnetic nanoparticles decreased in higher concentration of magnetic nanoparticle. Table amounts demonstrates an increase in mechanical strength and stiffness of the scaffolds by distributing the magnetic nanoparticles in the polymer matrix. One moment, please. Excuse me. I'm sorry. Okay, let's continue. As I'm at home and I have a small baby daughter, it happens in
Don't
online
worry.
congress.
It's okay.
In this slide, you see the MTT assay and the real-time PCR results. In the left side of the slide, you see that in MTT assay, the PLGA containing 10% magnetic nanoparticles showed the highest cell compatibility in comparison with control group, and the lowest cell compatibility was shown with PLGA with 0% magnetic nanoparticles. And in real-time PCR results with 3 neural markers, MAP2 as a neural markers had not a significant, showed no significant difference between the scaffolds containing 5% and 10% magnetic nanoparticles, while NSE and beta-tubulin markers show a significant higher, both show a significant higher expression in scaffold containing higher concentration of magnetic nanoparticles. And among immunocytochemistry analysis, you see the figures of expression of TUJ1 and NSE in all 3 samples, which in the left side of the slide, you see the expression of TUJ1 with green color, NSE with red color, DAPI in blue, and merged cells in the lowest row of the slide. As you see, the expression of TUJ1 and NSE increases while the percent of concentration of magnetic nanoparticles increase from 0 to 5 and 10%. And in the last image in the bottom of the slide and the right side, you see the highest expression of TUJ1 and the NSE marker in mesenchymal stem cells, which were seeded on 10% magnetic nanoparticle scaffold, as you see in this slide. And as a conclusion, in this study, we saw that magnetic nanofibrous PLGA scaffolds were successfully fabricated through electrospinning technique and do you hear me? I do not see my slides now.
We hear you.
Okay.
Don't worry. You have just 2 pages and it's not a matter.
Okay. And we think that the biochemical, mechanical, and mechanical properties of the scaffolds can be enhanced using the magnetic nanofibers and nanoparticles, as well as cell biocompatibility. And mesenchymal stem cells are one of the stem cells as a model, which can be replaced with other stem cells like induced pluripotent stem cells for other investigations in order to study further evaluations for spinal cord injury repair. Thank you for your attention, and I'm waiting for your question.
We thank you.
Evet Damla Hocam, geçelim mi bir sonraki sunuma?
Geçelim hocam. Soruları en sonda size de uygunsa.
Tamam, uygundur. 3. sunum için Hülya Yılmaz hocamızı davet ediyoruz.
Yes, I'm here. Greetings, dear participants, and I shall my presentation. Yeah. Is my screen visible?
Evet hocam, görüyoruz.
Someone,
Okay,
I think, mistakenly pointing out with red. So if somebody use it, please do not that. Yes,
Oh.
please. I turn it off, then you will start it again.
Okay.
Sistemsel olabilir Damla hocam o.
Sayın hocalarım, kimisi bazen yanlışlıkla basıyor. Hani mouse'a da dikkat edelim.
Ha.
Teşekkürler.
Tamam.
Evet hocam, tekrar açın isterseniz.
I opened again. And do you see on my
Evet
screen?
hocam. Hem
Okay.
görüyoruz slaytınızı hem sizi duyuyoruz. Buyurun.
Okay. I introduce myself. I completed my PhD education fundamentals of nursing department. I continued second PhD education translational medicine. I have been working as a lecturer at a high sciences faculty. And let's start. So the name is our project, investigation of the release of growth factors from nanofiber metloaded with apheresis plated concentrate. This project was implemented by Bursa Uludağ University Scientific Research Projects Coordination Department with a financing of 19,000 Turkish liras. Platelets are one of the peripheral blood components and are blood cells without a nucleus. Platelets are involved in a blood coagulation during hemostasis. Platelets contain many proteins, cytokines, and bioactive factors that initiate wound healing and regulate basic reactions. Membrane-bound alpha granules in the structure of platelets make intracellular storage, producing platelet-derived growth factor, transforming growth factor, epidermal growth factor, vascular endothelial growth factor, which are vital for wound healing. On this slide, we see the cells from which growth factors are sourced. At the same time, we see the functions of growth factors. APC is obtained by separating only platelets from donors, thanks to apheresis devices in the blood bank and spatial sets. Cells are separated from the donor in an anticoagulant solution and separated by centrifugation according to density and other filtration parameters. And other blood products are returned to the circulation after the selected product is separated. It takes 1 to 2 hours on average. It is a thrombocyte suspension obtained from a single donor. In recent years, in studies for applications such as tissue engineering and controlled drug release, nanofiber mats are preferred to be used as wound dressing due to their many advantageous properties. Especially electrospinning nanofiber surfaces, its hemostatic properties are similar to the morphological properties of the extracellular matrix of the skin in terms of moisture absorption, breathability, and providing suitable conditions for wound healing. Loading biomolecules, therapeutic agents, and various active substances on nanofiber surfaces can improve cellular behavior and promote wound healing and skin regeneration. When the literature is examined, no study has been found on nanofiber mats containing APC from the database accessed. However, there is a limited number of studies on platelet-rich plasma, abbreviation PRP, added nanofiber surface production models. The reason why platelet apheresis will be used in study is that platelet concentration ratio is higher than PRP. PRP is an autologous clinical treatment proven to accelerate wound healing. It is used in various clinical applications such as orthodontics, osteogenic care, orthopedics, acute and chronic wounds, and cosmetics. Biomedical applications of nanofibers produced by electrospinning. One of the most interesting and promising areas among the biomedical uses of nanofibers is wound dressing. Studies have shown that wounds that are kept moist can heal faster, but accumulation of exudate under the wound dressing can cause infections. In addition, with its controlled water vapor loss and excellent oxygen permeability, it provides the appropriate environment for wound healing by keeping the wound environment moist and getting oxygen, and creates an effect that accelerates wound healing. This study was conducted to investigation of growth factors released from ludids with APC composite nanofiber mats. For this purpose, the surface morphology of the composite nanofiber mat loaded with APC on one of its layers was investigated by SEM imaging technique. Then the time release of growth factors from the nanofiber surface was determined using the ELISA method. Tools and devices we have seen before slides. And the first stage, obtaining apheresis-related concentrates. APC to be used as an additive was obtained from a healthy and voluntary 31-year-old male, 178 centimeters, 84 kilograms, West Asian researcher who was part of the project team. Each APC was divided into aqueous volumes and then frozen at -18 degrees Celsius using 6% dimethyl sulfoxide. APC were taken to the textile engineering department and the nanofiber surface development process starts. Total protein and growth factors contained in APC were measured by BCA analysis and ELISA kit respectively. Second stage, production and morphological characterization of APC-loaded composite nanofiber mats. For the preparation of three-layer composite nanofiber surface, three different electrospinning solutions were first prepared. Electrospinning solution for the production of nanofiber surface that will from the primary outer layer, polyurethane polymer was prepared in dimethyl formamide at a concentration of 10% by mixing in a magnesium stirrer at 45 degrees Celsius for 24 hours. Electrospinning solution for the production of nanofiber surface that will form the secondary layer. The polyvinyl alcohol polymer was prepared in pure water at a concentration of 10% by mixing in a magnesium stirrer at 19 degrees Celsius for 12 hours. After the polymer is completely dissolved, 1 milliliter of APC will be added to 9 milliliter of polyvinyl alcohol solution and allowed to the mix at a room temperature. Electrospinning solution for the production of nanofiber surface that will form the inner layer. Polycaprolactone polymer was prepared at a concentration of 10% by mixing in a magnesium stirrer at 30 degrees Celsius for 12 hours. BCA measurement was performed with the protein assay kit as recommended by the manufacturer. When copper sulfide is added to the BCA solution used in the determination of BCA protein, a green color is formed as a result of the reaction. When protein is added to this solution, copper plus 2 ions turn into the copper plus 1 ions as a result of interaction with peptide bonds. At the end of the reaction, the green color turned into purple. After expiration, it was analyzed in a spectrometer at a wavelength of 562 nanometers. The effects of growth factors released from epithelial composite nanofiber met into PBS at a certain time intervals. These are epidermal growth factor, basic fibroblast growth factor, and the last platelet-derived growth factor. Quantitative analysis ELISA-kit was performed as recommended by the manufacturer. We have seen on the slide the results of morphological characterization on nanofiber surfaces and scanning electron microscope. Generally, the diameters of polyurethane nanofibers forming the primary layer range between 700 and 900 nanometers. And the statistical analysis, when the amount of release was compared between the FGF, EGF, and PDGF groups, as a significant difference was found in all time periods from the 48R to the 336R. When the time-dependent oscillation changes within each group were examined, there was no statistically significant difference in the FGF group, but a significant differences was observed in the EGF and PDGF groups. In the comparison made for PDGF, it was observed that there was a significant differences between 48R and 192 and 288 Rs, and the lower release occurred at these Rs. As a conclusion, the fact that APC-loaded composite nanofiber mats will be considered as a one arising in further studies constitutes the original value of this study. Thank you. And any question?
Please, thank you. Questions are at the end of the session. If there is any question, please type to the chat box. Also, you can write your email address to the chat box if you wish. Sayın hocalarım, isteyen hocalarımız mail adreslerini yazışma kısmına da yazabilirler. Buyurun Bekir hocam.
Evet. Sunum için teşekkür ediyoruz Hülya hocam sizlere. Dördüncü sunum değil, beşinci Özlem Özden Akkaya hocamız.
Hello.
Hello.
Yes. Do you see my presentation?
Evet hocam.
Okay. Hello, participants. Welcome to my presentation. Today I will mention you about explant culture of adipose tissue.
Özlem hocam, mümkünse kamerayı da açın isterseniz hocam.
Okay.
Teşekkürler.
Görebiliyor musunuz Damla hocam? Şu anda.
We can
Evet.
see. Everything is
Okay.
perfect. Your presentation and also you. Good
Okay.
luck.
Thank you. Today I will mention you about explant culture of adipose tissue. Full ekran yapmak istiyorum ama olmadı. Bu şekilde devam edelim. Sorry. Okay. Değiştiremiyorum. Siz görebiliyor musunuz? Şu anda
Hocam, sadece ilk ekranı görüyoruz. Elle isterseniz yandan değiştirin. Manuel
Tamam.
olarak. Üstlerine slaytların tıklayarak hocam.
tamam,
By
bu
clicking
şekilde.
manually with the slides.
Okay. Stem cell therapy is considered as a beacon of hope in many incurable diseases, and stem cell is used in the treatment of various diseases in medicine and veterinary medicine. Despite of being very common in pets, animals, and equine, in the field of veterinary medicine, stem cell-based practices in the bovine yet remain very limited. Mesenchymal stem cells are multipotent cells that can be obtained from many tissues of mesodermal origin. Although bone marrow is the most widely used source of the stem cells, the fact that it is a painful and laborious method to obtain, the station paved the way for finding new stem cell sources such as adipose tissue. Adipose tissue-derived stem cells showed multipotent properties as they are able to differentiate into both osteogenic, adipogenic, and chondrogenic lineage. The reports about the expression of pluripotent stem cell markers in bovine adipose tissue stem cells have been published earlier on. For example, SOX2, NANONG, and OCT3-4. However, adipose tissue less immunogenic than bone marrow. Stem cells derived from adipose tissue can be obtained as enzymatic method or explant culture method. In the studies, it has been reported that obtaining cells in explant culture is fast and economical, as well as the rates of proliferation and viability of the obtained cells for explant culture compared to the enzymatic digestion method. In the study, it was aimed to obtain stem cells from bovine adipose tissue by explant culture method. The use of slaughterhouse material in the study eliminates ethical problems and also provides an advantage in terms of amount of tissue and cell to be obtained from cattle. Adipose tissue were harvested aseptically from 1.5 to 2 years old male cattle from the local tower. If you are going to work with slaughterhouse materials, you should be more careful when collecting tissue to be aseptic. Subdermal adipose tissue samples were collected into sterile tubes which contain sterile 0.9% sodium chloride solution with 2% penicillin streptomycin and 0.2% amphotericin B. Samples were brought into lab within 15 minutes post-collection. Samples were brought into the laminar flow and betadine sodium chloride secule suspension were used for proper disinfection of tissue. Disinfection steps is very important. After sterilizing, samples were suspended further in sodium chloride and antibiotic mixture in order to clear betadine. Other lining of the tissue was discarded and tissue samples were mechanically split into smaller parts. Yes, there are 3 pictures. First, fresh adipose tissue in sodium chloride contain antibiotics and antifungal. Second and third pictures are disinfection stage with betadine. The remaining tissue parts were seeded into well plate with medium supplementation and incubate at 37 centigrade degree in 5% carbon dioxide. Cell culture medium consisted of 10% fetal bovine serum, 1% penicillin streptomycin, 0.1% amphotericin B, and 200 millimolar L-glutamine in low glucose alpha men. Firstly, outer lining was discarded. Then small pieces were cut from inner area, inner parts. Tissue pieces were incubated in incubator. You can see at second picture. The tissues weren't touched for about a week. The tissues weren't moved. Thus, tissue adhesion of the tissue was ensured at plate. After adhesion, the medium was changed in every 3 days. Mesenchymal were isolated by Explant culture method about 3 weeks. At 18% confluence, cells were gently harvested and passage. I said that after seeding, stem cells were isolated in 3 weeks. The cells had fibroblast-like morphology. After first passage, most of the cells showed fibroblast-like morphology too. We can see excretion of stem cells here. In the second picture, we can see cells increasing around the tissue. First picture is passage 0, second one, passage 1. In both pictures, we see that the cells have fibroblast-like morphology. Explant culture method is believed to be an easy and more economical way to harvest stem cells. I just told you about cell isolation, but this work continues. In the further genetic and molecular characterization of the isolate cells, along with multilineage differentiation, studies are planned. This photograph is the shadow of the opium castle. I want to share with you. Thank you your attention. Thank you for your attention.
Evet hocam, teşekkür ediyoruz sunumunuz için.
Thank you.
Damla hocam, diğer Maksut'a geçelim isterseniz.
Tabii hocam, nasıl uygunsa size. Buyurun.
Evet, dear Maksut, we are listening to you.
Yes, yes. Are you seeing me? Can you see my?
We can see you, but can you please yeah, it's coming now.
Evet.
Yes, it's okay.
It's okay now, but please do not make full screen because it is stacking, and please change
Okay.
your presentation by manually.
Slides.
Please, can you try for changing the pages now?
Yes.
Yeah, it's okay.
Yes. Good afternoon to everyone. Distinguished Congress Committee of this great event. Thank you for giving us this opportunity. Let me introduce myself. I'm coming from North Macedonia, and I'm a veterinarian and PhD student. Different from your thesis, I'm working on commensal E. coli from gastrointestinal tract, and my thesis is isolation of commensal E. coli, who is resistant to third generation of antimicrobials. And so in our University of St. Cyril and Methodius in Skopje, we had a doubt what to study, and that's, in one hand we have commensal E. coli, and the other hand we have antimicrobials, and that's cephalosporins. We are focused in cephalosporins resistance, and I can say cephalosporin resistance, classified from World Health Organization at the critical important antimicrobials, and a large amount of antimicrobials are used in animal, and it's expected to be growth for 11.4%, and there are a great number of antimicrobials who are overused and misused, and we choose, in other hand, commensal E. coli because she's a great and a large pool of resistant genes on her home. That's why we did this comparison, and we are reference laboratory, and we work according to European Food Safety Agency, and we do proficiency testing, and E. coli is among them. It's mandatory. So let's start my presentation. It's a very small time. I eliminated a large of slides to be in 5 minutes, and so I'll try to be in that range of time. The aim of this research is to detect and prevalence the resistance commensal E. coli from cow manure in dairy cow, and to determine the minimum inhibitory concentration of those isolates. And the third aim was to determine the phenotypic properties of those slides. So I'm working over the 20 years like veterinarian in a border with Albania, and we did choose a wide spatial range of farms that they overuse the antimicrobials. The farmers is using by themselves. It's a large number of black market and the easy access of antimicrobials from the farmers and from professionals. That's in a human medicine and veterinary medicine. That's why it's a great issue of top 5 issue of the world, antimicrobial resistance. And so we did a methodology, the first step of detecting the prevalence, and normally as a reference laboratory, we working through decision of European Union 2013-652. That's the manual for reporting and surveillance of antimicrobial resistance, and this is the protocol of DTU that is giving us the coordinates. So materials and methods, we did it normally from the protocol and the regulation of taking the samples of biological samples, 882.2004, and we strictly adhere to the cold chain temperature from taking samples to the reference laboratory. And normally it takes, we did 159 samples from dairy cow, and we have 34 included farms in this research. Normally, I don't want to explain much in details how did we selected those farms and which kind of antimicrobials they use. There's a questionnaire over each farms and the farmers. So the first step was detecting and identification of isolate. As you can see in the plates, that's the first plate. There's containing cephaloxime. That's the 3rd generation of cephalosporin, and the second plate here is a highly differential plate. That's E. coli, Escherichia coli, and mostly of the isolates we did biochemical methods with Vitek. That's a big machine who works in several biochemical tests the isolate and confirm that we have commensal E. coli. That's the clearly isolate. And so to go in a second aim and to show determine the minimum inhibitory concentration of those range of antibiotics, cephalosporins, and other groups of cephalosporins and antimicrobials, we used international standard according to European Union 2007-76. And why we did this method, it's the greatest method. It's a great number of antimicrobials and a wide range of concentration of these microbials. And we have in the first plate, we can see in the next slide, we have in this slide, we have in the left one is the EUPSEC1 plate. We worked in a 2 plate. In the left side is the EUPSEC1 plate. And there's 14 different antimicrobials. And the right plate, that's EUPSEC2 plate, it's 10 different antimicrobials, and a larger number, including cephalosporin of third, second, and fourth generation. I don't want to take time in the range of antimicrobials. I want to be exactly in time, and so I just want you to see that as this you can see in cephalotoxin, that's in a third rate, is 38 of isolates from all 39 isolates. We have a resistance, and that's a very serious thing. And ceftazidime, the second cephalosporin representatives of the third generation, we have also 38 isolates from all 39 isolates. That's 97.4 of isolates. And we, according to DTU of Danish Technical University, we interpreted it through eco values, that epidemiologic cutoff value, not clinical cutoff value. So every single isolate that is resistant to more than 1 milligram per milliliter to cefotaxime, ceftazidime went further to the second plane. And this is the second plane. It's a second plane. I just want to potential in a few words that there's 6 different antimicrobials only from cephalosporin group, and which is second, third, and fourth cephalosporin. And we have very strange and very important results. As you can see, we have 100% resistance to cefotaxime, and ceftazidime. That's 3 groups of antimicrobials. And it's interesting because if those cephalosporins are in combination with beta-lactams inhibitors, which is in the case in the clavulanic acid, we have no resistance. It's resistant 0%. So in conclusion, we from 34 farms, we detected 11 farms positive in cephalotoxin resistant bacteria, and resistant were from 100%. We had resistant to cefepime, cefotaxime, and ceftazidime, and no resistant to, as I mentioned before, with combination with beta-lactam inhibitor. And the third aim was detection of phenotypic properties. I just deleted that kind of slide. There's a scheme of how it detects, and from results from 36 of isolates were extended spectrum beta-lactamase, and 1 isolate was an AMPC phenotype. And so this research is still ongoing. We are now taking place of PCR, conventional method of PCR to detect the resistant genes that all those isolates have. And so I will finish with Sir Alexander Fleming. That's the person who's playing with penicillin treatment, and this is a very, very Accutane is morally responsible for the death of the people who are succumbed to infection from the penicillin resistance. So we have to take care for using and overusing antimicrobials. There is a great resistance over the world, and the more we use, the more we lose antimicrobials. And thank you for your attention, and thank you for being part of this great Congress. And I'm happy to answer your question. I will leave my email, and I will be happy here to answer your questions. Thank you.
Thank you, dear Maksud, for your nice presentation. Nice and very comprehensive. Good luck.
Thank you. Thank you very much.
Bekir Hocam buyurun.
Evet, bir sonraki sunumumuz için we are waiting for Trichomonas fetus presentation. Şu anda.
Yes, good afternoon. Okay, I'm in the middle of this process and hope that everything will be okay. And I'll click here. Okay, hope that, can you see my presentation? You see my presentation, okay. Okay, great. So once again, good afternoon, ladies and gentlemen. My name is Joanna Dąbrowska, and today I would like to talk to you about comparison of 4 commercial methods for the extraction DNA by 2 molecular methods for Trichomonas fetus DNA identification in spike filling pieces. Trichomonas fetus is a single-celled protozoan parasite. The microorganism is 525 micrometer in size and is spindle-shaped with 4 flagella with an undulating membrane. Under the microscope, we can see movement of this parasite and is jerky and in forward direction. Trichomonas fetus is reported as a pathogen of the large intestine of cats and colonize the ileum, cecum, and colon. The presence of Trichomonas fetus in cats causes chronic large bowel diarrhea, and parasites in cats are found worldwide with prevalence around 59%. According to manual OIE, diagnosis of feline trichomoniasis is based on fecal examination with using generally cell cultures and PCR tests. Pieces are identified as practical samples for molecular diagnosis of infectious disease, including trichomoniasis. However, extraction of high-quality DNA from fecal samples can be quite challenging because of the presence of PCR inhibitors and that are co-extracted with DNA. So the aim of our study was to compare the efficiency of 4 commercial kits for the isolation of Trichomonas fetus DNA from cat pieces by 2 molecular methods. So here you can see these kits which were compared in our study. And firstly, we cultivated Trichomonas fetus cells in diamond medium, and then we used Neubauer chamber for counting cells. Fecal samples from clinical healthy cats were enriched with an appropriate number of cells. A series of dilutions corresponded to 10,000, 1,000, 100, 10, 1, and 0.1 Trichomonas fetus cells per sample. Each variant we prepared in 6 replicates. The efficiency of the isolation kits we evaluate by 2 molecular methods, which were PCR according to Felenstein, and also our own developed method LEND. Here we have detailed results from our study. And on this slide, we can see summary results of these examinations and the best extraction efficiency produced by ZR fecal DNA mini prep kit and allowed detection of equivalent of 0.1 parasite by lamp in 33.3% results. QIM DNA stool was slightly lower isolation DNA efficiency with sensitivity 1 cells in 66% results in lamp and 33 in conventional PCR. Furthermore, surprisingly, Sherlock AX has also relatively high efficiency. LAMP was able to detect Trichomonas fetus cells on the level of spiking 10 cells with 60%, and the least sensitive method was ultra clean fecal, which could only detect 100 cells in 33% by LAMP and also 100 cells in 16.6% results by PCR. And as you can see in this slide, in the table, performance of Z lamp was statistically significant better than S PCR and U PCR. Furthermore, statistically significant differences were found between performances of U lamp and Z lamp, as well as between U PCR and Z PCR. In conclusion, our investigation showed the highest efficiency ZR fecal DNA mini-prep extraction kits dedicated for fecal samples in Trichomonas fetus detection by lamp. The second method among compared extraction kit was QIMP DNA stool, and Sherlock AX has the worst efficiency isolation DNA from Trichomonas fetus with both molecular methods, and the least was ultra-clean fecal. Furthermore, it is worth to notice that the first time efficiency of extraction methods was evaluated by lamp. Thank you for your attention.
We thank you.
Damla Hocam,
Buyurun hocam.
Joanna Hanım benim meslektaşım sayılır. İkimiz de parasitoloğuz.
Evet.
Ben birkaç bir şeyler söylesem de siz çevirebilir misiniz?
Tabii ki hocam, buyurun. The moderator, you are mostly working same department, or he is also parasitolog, Joanna? Can you hear us? Joanna is connecting again.
Can you hear me?
Joanna?
Yes.
Yeah.
Yes.
Associate Professor Bekiroz is going to ask you a question, if it is possible for you.
Yes, okay, of course.
Evet, sunumunuz için tekrar teşekkür ediyorum.
Thank you for the presentation.
Trichomonas fetus, biz bu paraziti sığırlarla tanıdık, sığırlarla biliyoruz.
Do you know
Ama,
this Trichomonas parasite from bovine?
ama son yıllarda yapılan çalışmalar bu parazitin kedilerde olduğunu da gösteriyor.
The last research shows us that this parasite can be originated from the cats.
Yes.
Acaba sığırlara bulaşmada kedilerin son konak olduğunu söyleyebilir miyiz? Belki ileriki çalışmalarda ortaya çıkar.
Kediler mi son konak olabilir
Evet,
hocam?
kediler son konak olabilir mi? Yani bu zamana kadar sığırlarda böyle bir durum söz konusu değildi.
Is it possible that if the cats are the last host, what do you think he is wondering?
Okay, thank you for this question. So the situation with Trichomonas fetus is slightly complicated because this parasite can reside in the three hosts, which are cattle and cats and also pigs. And there is a
Domuzlarda da hocam.
Domuzlarda da evet bulmuşlar, evet.
Yes, and there is a many hypotheses, not maybe many, but there is a hypothesis that there is a one parasite, but also the newest studies which we conducted showed that in cats, that these parasites are not in the same species because they can reside in cats' intestinals, and they give
In cat or in pig? Which species intestine?
In pig, yes, in also, but there is only commensal. It's not affects pigs with disease symptoms is only commensal, which
Domuzlarda komensal olarak bağırsakta da bulunabiliyormuş hocam.
Ha.
Yaptıkları çalışmada.
Bu son kana kolan iç durumla ilgili ne söylediğini tam olarak anlayamadım bu sesle.
What do you think about the last host? Which
Yani
mechanism
kediden sığıra bir bulaşım. Bulaşma,
okay.
transmission.
can cat infect the bovine or cattle?
Yeah, yes, now I understand what you mean. These parasites can live only in one host. There is no transmission between this host. There is one definitive host, let's say.
Tek bir tane host oluyormuş, birbirlerinden geçiş olmuyor diyor hocam.
Birbirlerinden geçiş olmuyor. Aslında bu konu çalışılmaya açık bir konu. Şimdi eğer kedi dışkısıyla sığır olursa, buluşursa, yani kontamine olursa, kedi dışkısında Trichomonas foetus var diyelim. Sığır da işte otlarla vesaire şeyle bunu kontamine olmuş bu otları yer ise enfeksiyon ulaşabilir. Biz bu zamana kadar koitus yoluyla, yani işte bu erkek sığırlar portördür. Bunlar çiftleşmeyle, erkek sığırlarda hiçbir patojenitesi yok. Çiftleşmeyle sığırlara geçer. Sığırlarda da bu gebelik abortlara sebebiyet veriyor bu hastalık. Biz hep böyle biyolojisini bildik ve böyle şey yaptık. Ama son yıllarda mesela okuyoruz yani makaleleri. Kedilerde bulunuyor mesela.
Evet, ama tek host olduğunu söylüyor. Yani hani
Evet.
geçişin olmadığını.
O biraz böyle biyolojiyle beraber çalışılacak veya işte kedide izole edilenle sığırda izole edilen genetik olarak yakınlıkları. Şimdi PCR çalışıyor ya.
Evet.
Onlar işte bakılmalı. O şekilde belki bir ileriye dönük bir sonuç çıkabilir. Toparlamak gerekirse anladık yani şu durumda onun son konak olduğunu söyleyemiyor. Tek bir konağın olduğunu söyleyebiliyor.
Evet.
Kendisine başarılar diliyorum bu konuda yaptığı çalışmalarla.
Thanks very much. We wish you all the best for your works. Also, it's a really good topic for working because it needs many research for checking.
Türkiye'de de kedilerde bulundu. Onu da söyle.
Tamam hocam. Even this needs many works for working on, for researchers, because there is many unknowns in it. Even people should, or researchers should work on its genetic and PCR.
Yes.
And also in Turkey, it has been found in cats. He's added this one in Turkey. Yeah.
Evet.
I think that I saw this publication from Turkey about this. So yeah, I am still in touch with this topic. So I think that I saw this. But yeah, that is very interesting.
Yeah. Var mı hocam başka bir şey?
Teşekkür ederiz. Şeyi bozmayalım. İnsicamı devam edelim.
Thank you so much, Joanna. See you later.
Teşekkürler.
Bye
Thank
bye.
you for
Good luck.
presentation. Joanna. Thank you.
Thank you.
Evet, bir sonraki sunumumuza geçelim. Safe diye herhalde söyleniyor.
Yes, can you hear me?
Yeah, we can.
Evet.
Okay, I will share my presentation. You can see my screen?
We can, but can you try it for changing the pages? Sometimes it is stuck, just we are seeing the first page. That's why yeah, we can see. Good luck.
Okay, thank you. So hello, everyone. I'm a PhD student from Morocco. So thank you for organizing this Congress. My presentation is entitled Theoretical Prediction of Staphylococcus aureus and Pseudomonas Aeruginosa Adhesion on 3D printing filament materials. So this work was carried out in the laboratory of microbial biotechnology and bioactive molecules, Sidi Mohammed bin Abdullah University in Fez in Morocco. So in natural environments, microorganisms are most often attached to solid surfaces and are generally provided with sufficient nutrients to ensure their viability and growth. So this phenomenon is called biofilm. Biofilm formation can be divided into 5 stages. The initial contact of the moving planktonic bacteria with the surface is the starting point, which is still reversible at this stage. The bacteria will then start to form a monolayer and will produce an extracellular matrix for protection. In these stages, the formation of microcolonies takes place, which exhibit significant growth and cell-cell communications such as quorum sensing. So the biofilm grows in a 3-dimensional manner, and the attachment is now irreversible. In the last stage, some cells of the mature biofilm start to detach and disperse into the environment as planktonic cells again to potentially start a new cycle of biofilm formation. Rather, microorganisms have been shown to naturally accumulate on a wide variety of surfaces. Those surfaces include household and industrial pipes, plants, and animal tissue, biomaterials such as contact lenses, medical devices, including implants and urinary catheters. So in the absence of hygiene measures, this medical equipment in contact with biological fluids will be exposed to many microorganisms and micromolecules. These different entities can adsorb or adhere to a surface and colonize it. When the microorganisms involved are pathogenic, the dangers to human health are many, from intestinal infection caused, for example, by Salmonella, to nosocomial infections, and therefore a means of multi-resistant pathogenic germs transmission. Then the aim of our study is to study the prediction ability of Staphylococcus aureus and Pseudomonas aeruginosa as nosocomial pathogens to adhere on 4 filament materials, nylon, ABS, PUT, and PLA. Their materials were chosen for their low cost, good strength, multi-material capability, and large using in medical equipment. So for materials and methods exploring, a desktop 3D printer was used to fabricate the test materials. The printing parameters such as printing speed, printing angle, and temperatures were controlled using 3D printing software. And all materials queued into 1 square centimeter were soaking in ethanol for sterilization and rising 3 times with distilled water. After that, the bacteria were grown in Loria bertani medium, and the cells were collected by centrifugation, and the bacterial surface was prepared by depositing on filter paper using negative pressure to create a smooth and flat lawn of compactly organized cell. After that, for contact angle measurements, the sessile drop technique was used to evaluate the physicochemical parameters of bacterial and substrata surfaces using a goniometer coupled to a video camera system and desktop computer. Then for 3 liquids, 3 contact angle measurements were made on the surface using water, formamide, and diodomethane. The Lifshitz van der Waals compound, the electron acceptor and electron donor properties of material surfaces were estimated by Van Os approach. So as a result, the contact angle with water, generally the contact angle with water, it's called theta water, it's the first column, was applied as a qualitative indicator of hydrophobicity of the material surface. So the surface is considered hydrophobic if this theta water is higher than 65 degrees, and delta GIWI is less than 0. And in fact, the nylon, ABS, and PUT surfaces were qualitatively and quantitatively hydrophobic, but on the other hand, the polylactic acid surface showed qualitatively hydrophilics and quantitatively hydrophobics. But all filament materials had a higher electron donor compound than an electron acceptor compound. But in terms of bacterial strain, physical properties, the results show that S. aureus and Pseudomonas aeruginosa are qualitatively and quantitatively hydrophilics, and they were many electron donor. That's about just the physicochemical parameters of substrata and bacteria. Now we are going to to study the ability of adhesion. So to anticipate bacterial adherence to surfaces, several theories been proposed. The Van Os theory was used in this study because it showed consistent findings with microbial adhesion. So as a result, total interaction-free energy calculations for the bacterial adhesion process on filament materials were conducted. So the total free energy between S. aureus, for example, and PUT is negative, indicating thermodynamically favorable adhesion. And in the case of nylon ABS and PLA surfaces, the total free energy is positive. Furthermore, Pseudomonas aeruginosa shows unable to adhere to nylon and PLA, which values of total free energy are positive. Contrary-wise, this adhesion is favorable for ABS and PUT with also a high values of delta GEB, which confirm the importance of acid-base interaction in the bacterial adhesion process. Then, nylon and PLA presents the suitable materials that the adhesion was unfavorable for both bacterial species, but in general, when strains are evaluated in ideal growth conditions, experimental investigations on bacterial adherence to abiotic surfaces are required. However, bacteria in nature and or in synthetic ecosystems are exposed to a variety of environmental stresses and a complex phenomenon influenced by a number of factors which may affect the physicochemical properties of cell surfaces and as a result bacterial adhesion behavior. So thank you for listening. Wish you a life full of love, science, and success from Morocco. Thank you.
Evet, thank you for presentation. Damla Hocam, diğer sunuma geçiyorum. Duygu Güleç Satır Hocam.
Yes, hello, dear researchers. Merhabalar herkese.
Herkese merhabalar. Bizlerden de merhabalar.
Ekran paylaşıyorum şu an.
Buyurun hocam.
Can you see my presentation? Görebiliyor musunuz hocam?
Evet, görebiliyoruz.
Tam ekran yaptım şöyle. Ama ilerlemiyor zannedersem.
Tam ekran oldu şu anda. İlerlemiyor mu?
Durdurdum. Tekrar paylaşıyorum.
Tamam.
Biraz bilgisayar yavaş. Hani sorayım.
Şu an bilimsel program gözüküyor Duygu
Evet,
hocam.
pardon. Durdurup tekrar paylaşacağım.
Tamam.
Saniye. Evet, geldi zannedersem değil mi?
Evet, şu an geldi. İsterseniz tam ekran yapmadan.
Evet. Evet, teşekkürler.
Tamam.
I am Duygu Güleç Shutter. I work in Ege University Faculty of Nursing, Department of Women Health and Diseases Nursing. The title of research is Examining of Fear of Birth and Parental Attachment in Expectant Mothers and Fathers. Introduction. Although pregnancy is a physiological experience for women, physical and physiological change can cause anxiety and fear of childbirth in both mother and father. Parental attachment is defined as the relationship and interaction that develops between parents and fetus during pregnancy. Aim of the research is to examine fear of birth and parental attachment in expectant mothers and fathers. Materials and methods. The descriptive and cross-sectional research was carried out in Izmir. The population of the study consists of expectant mothers and fathers who applied to obstetric polyclinic of a training and research hospital on this specific date in 2020 year. Participants. The sample of the study consisted of a total 288 participants, 202 of which were mothers and 86 fathers. Inclusion criteria for the study were defined as being literate, being over the age of 18, being in the second or third trimester of pregnancy, feeling the baby movements, those with health problems in the fetus, multiple pregnancy, and psychiatric disorders diagnosed in expectant mothers or fathers were excluded from the study. Data collection. The purpose of the research was explained to the mothers and fathers waiting in the line for control in the antenatal clinics of the hospital where the research will be conducted, and the volunteers were asked to fill in the questionnaires themselves. For mothers, questionnaire forms introductory information for mothers, visual analog scale to evaluate fear of childbirth, and maternal fetal attachment scale to evaluate parental attachment were used. As for fathers, introductory information form for fathers, visual analog scale, and paternal fetal attachment scale to evaluate parental attachment were used. Data analysis, descriptive analysis were conducted to determine the introductory characteristics of mothers and fathers participating in the research. The relationship between the variables was tested with parametric and non-parametric tests. The simple linear and multivariate linear regression analyses were implemented for independent variables affecting parental attachment in expectant mothers and fathers. Results. Mean age of expectant mothers was 28. Mean age of expectant fathers was 33. Most of them university graduates and employment. 58% of mothers and 57% of fathers' wives were multipares. Results. Scores of fear of birth. The mean fear of childbirth score evaluated by visual analog scale of expectant mothers was 5.13. As for expectant fathers, it was 5. The difference was not found to be significantly. Results. Cause of fear of birth. While the mothers have mostly fear of birth pain and the healthy of the baby, the fathers specify that they have fear about their supposed pain and health. Results. Attachment scores. Fetal attachment scores of expectant mothers were 96 and fathers were 92. The scores of mothers were found to be significantly higher. Parental attachment scores by descriptive characteristics of expectant mothers and fathers. Attachment scores of mothers with low education level, multipares, and unplanned pregnancy and fathers with low economic status were found to be lower. There was no significant relationship between birth fear scores and attachment scores of expectant mothers and fathers. Regression analysis results. In the regression model, parity and planned pregnancy in expectant mothers explain 8.4% of the model, and economic status explains 7.3% of parental attachment in fathers. As a result of research, it was found that the birth fear levels of mothers and fathers were similar. It was concluded that mothers had the most fears related to birth pain and the baby health, while fathers had fears related to their supposed health. Another result of the research is that mothers' attachment to the fetus is higher than of fathers' attachment. Thank you for your attention.
Evet Duygu Hocam, teşekkür ediyoruz sunumunuz için. Şahin Hocam'la devam ediyoruz. Şahin Çakır Hocam.
Tamam hocam, buradayım.
Buyurun
Merhabalar.
hocam. Merhabalar.
Duygu hocam. Hocam geldi sanırım.
Evet, sunumunuzu görüyoruz hocam.
Kırı taşıdı. Değişimde oluyor sanırım.
Hocam tam ekran yapınca bir sıkıntı oluyor ama bir deneyin isterseniz.
Şu an değişim oluyor mu hocam?
Olmadı. Olmadı hocam.
Bir alayım hocam.
Ha, şu an oluyor. Değişim.
Şu an oluyor. Tamam.
Evet.
Dear Chairman and participant, my name is Şahin Çakır. I am a PhD veterinarian. I have been working Ministry of Agriculture and Forestry, General Director of Agriculture Research and Policy, Department of Animal Health, Food and Feed Research. My presentation title is Epidemiology of Bovine Tuberculosis from past to present in Turkey. You can see the presentation on this slide. I would like to give short information about bovine tuberculosis. Bovine tuberculosis is an infectious and chronic disease that spreads worldwide. Mycobacterium bovis has the widest host range among Mycobacterium tuberculosis complex members, transmitted to animals mostly by Aureus. It's a socioeconomic disease that zoonoses, loss of yield, and trade restriction. Worldwide estimate over the 50 million infected cattle and 3 billion annual economic loss. It has more than 40 wildlife reservoirs. Mycobacterium bovis is responsible for about 1 to 15% of all tuberculosis cases and about 30% of children. 11,821 tuberculosis cases in 2017 in Turkey, at least 5.3% Mycobacterium bovis. Last calculated estimate socioeconomic impact of bovine tuberculosis is between 15 to 59 million dollar per year. Ago 2 decades in Turkey, the number of bovine tuberculosis outbreaks in Turkey has increased more than 30 times in the last 2 decades. Story of bovine tuberculosis. Bovine tuberculosis originate in Europe and spread throughout the world during the colonial times. Koch, 1882 discovered the tuberculosis bacillus and subsequently developed tuberculosis in 1890. Finland, 1890 is first country to initiate bovine tuberculosis eradication campaign using tuberculosis skin test. Galmette and Guerin, 1913 developed the Mycobacterium bovis vaccine. Preferred protein derivative was produced by Siebert and Long. 1934, 1940, Wood et al. 1980 developed the interferon gamma press assay in Australia. Ottoman Empire period, slaughter should be done at the slaughterhouse in 1882. Tuberculin use started in 1891. The Institute of Bacteriology was established in Istanbul 1893. Tuberculin was produced in 1893. Legal registration was made in 1913. It was decided to give compensation for tuberculosis positive animals in 1913. Republic of Turkey period, the prevalence of bovine tuberculosis was found to be 5% in 1920. For the first time in Turkey, tuberculin was applied to 7,335 cattle in Istanbul, 1929, and 676, 9.21% reactor animals were detected. Tuberculin was administered to 43,439 cattle on the Eskişehir-Ankara-Çubuk line between 1937 1938. The second now-sounding study on the bovine tuberculosis at positivity was detected in 581, 1.33%, while 10% of reactor animals were found as a result of the tuberculin application in state livestock institution in the 1938. This rate decreased to 0.39% in 1962 as a result of the struggle. In 1946, 100% positive was detected in 92 dairy cows in 5 enterprises in Istanbul. This table shows 17 provinces in and the result of the light sounding work carried out in 1949 in Turgut Argun. Bovine tuberculosis prevalence was found to be 6%. In 1950, 79, 40% of 197 animals in 6 enterprises in Istanbul were found to be positive. In 1986, tuberculin skin test was applied to 249,387 cattle and a total of 1,983, 0.78% cattle were found to be bovine tuberculosis positive. In 1987, tuberculin skin test was applied to 63,920 cattle and a total of 252, 0.39% cattle were found to be bovine tuberculosis positive. In 2,522, 9.9% of 5,257 animals from different provinces were found to be positive. Finally, 2011, with the application of the tuberculin in 2,857 hertz on the national scale, the prevalence of individual cattle was reported as 1.4% and approximately 2.5% in cattle herds. We carry out the national project from OIE World Animal Health Information System and Ministry of Agriculture web-based recording system. When tuberculosis outbreaks reports were received, the response of official veterinarian in Turkey, 371 from 72 provinces, and breeders 217 from 61 provinces. Questionnaire were evaluated. Opinion of official veterinarian and breeders visit on site 7 regions, 14 provinces, 28 enterprises were complete. The slide shows distribution of bovine tuberculosis outbreaks last 16 years in Turkey. I would like to drive your attention on the increasing trend in the number of bovine tuberculosis outbreaks peaked 2018 years. You can the spatial distribution of the bovine tuberculosis outbreaks in the last 16 years according to province. Increase the risk socioeconomic loss, increase risk of food security, increase from wildlife in some countries. Although there is a decrease in human transmission with pasteurization of milk, in the instance of Mycobacterium bovis pediatric and extrapulmonary tuberculosis is high. Increase the multi-resistant Mycobacterium bovis strains. Increase the co-infection with HIV. As a result of this research, it was understood that 35 to 55 of the animals due to the tuberculin application in the outbreaks enterprises were slaughtered or died. This rate and information we have obtained from the field indicate that there is a significant national increase in the prevalence of bovine tuberculosis. In conclusion, conducted a pilot survey representing the national animal population with the tuberculosis screening to determine the current bovine tuberculosis prevalence show that it is important for the determination of the national fighting strategy. We must leave them healthy future. Thanks for your attention. And also I would like to thank the Congress organization team.
Evet Şahin Hocam, teşekkür ediyoruz sunumunuz için. Hiç bekletmeden bir sonraki sunuma geçelim. Kadir Akar Hocam.
Hello, Chairman, and hello, dear researcher, dear participant. I would like to introduce myself firstly. I have been working in Pandemic Veterinary Control Institute for 5 years in a national reference laboratory for brucellosis and I am PhD veterinarian. And today I want to present some information about Brucella field strain isolate in Turkey in 2021. I guess you see my screen.
Evet hocam. Sunumunuz gözüküyor. Full screen'de bir sıkıntı var ama hocam.
I was okay, I guess. Is it okay?
Evet.
Yeah, some information about Brucella field strains isolated in Turkey in 2021. As an introduction, brucellosis is a common zoonotic disease that is prevalent in Turkey as well as especially Mediterranean countries and in the world, and causing serious economic losses. The gold standard in diagnosis of Brucella is bacterial isolation, and strain have been identified at species and biovar levels by classical and molecular techniques according to the OE manual. The study of evaluating the Brucella species and its biovars circulating in the field have been conducting associated with mass vaccination programs starting in 2012 in Turkey. The aim of this study is to determine species biovar and geographical distribution of Brucella field isolates sent from Marmara region and other regional institute for biotyping to the Turkish National Brucellosis Reference Laboratory in 2021. As a material and methods, tryptic soy agar supplemented with heat inactivated bovine serum was used for all culture studies. The working petri dishes were...
Kadir hocam araya giriyorum ama slaytlarınız ilerlemiyor hocam.
Sorry.
Evet. Tıklayarak gidersek hocam sol taraftan.
But it's not full screen.
Materyal metotta kalmıştık. İsterseniz buradan devam edebilirsiniz.
Are you seeing?
Görüyoruz şu anda.
It's going forward, right? You see?
Görüyoruz hocam. Şu anda sıkıntı yok.
The working petri dishes were incubated for 4-5 days at 37 degrees with percentage 10 carbon dioxide added medium, according to Alton et al. And biovars of the isolates were investigated grow properties, carbon dioxide requirement, dehydrogen sulfide production, thionine, fuchsine, safranine, penicillin, streptomycin, and erythritol sensitivity, as well as leases with Tiflis, RC, and Isatnagar phages, and agglutination with monospecific sera A and M. Media containing streptomycin, penicillin, and erythritol were used to identify for vaccine strains, according to the OE manual. As a result, a total of the 401 isolates were collected in the reference laboratory in 2021. Of these isolates, 338 were from the cattle, 53 from the sheep, and 10 from goats. The sample belonging 5 regions in the Turkey were analyzed. Except Black Sea and Aegean Sea, we couldn't obtain. Distribution of biovars in a cattle: Brucella abortus bio 1, 3, 4, atypic Brucella abortus rough strain, and Brucella abortus S19 vaccine strain, as well as Brucella melitensis biovar 1, 3, and atypical Brucella melitensis. Normally we know...
Kadir hocam tekrar bölüyorum. Slaytı göremeyen arkadaşlar var. Evet, sol taraftan tıklarsak hocam. Evet.
Bu şekilde mi devam edeyim?
Aynen hocam, bu şekilde devam edelim.
Tamamdır. As normally we know the main hosts for Brucella abortus in the cattle, Brucella melitensis for the goats and sheep. But in this case, some cases, Brucella in the cattle, Brucella melitensis biovars and atypical Brucella melitensis detached in the cattle. And in the sheep, Brucella melitensis biovar 1, 3, atypical Brucella melitensis and Brucella melitensis Rev1 vaccine strains, as well as the Brucella abortus biotype 1. We see that the transmission is possible between cattle and sheep. In a goat, Brucella melitensis BioWar1, BioWar3, atypical Brucella melitensis, Brucella melitensis Rev1 vaccine strain, as well as Brucella abortus BioWar1 and BioWar3 were detected too. Strains obtained from the cattle, the most common strain is Brucella abortus biotype 3, percentage 74.6. From sheep, the most common strains is atypical Brucella melitensis isolates, percentage 51. And from the goats, the most common strains is Brucella melitensis biotype 3, were detected for percentage 40. We are seeing the table total number of abortus and melitensis isolates for in the 2021. As I mentioned, in the cattle, you are seeing for the Brucella abortus biotype 3, 252, and you are seeing in the cattle Brucella melitensis isolates and atypical. For the sheep, you are seeing normally you have to see Brucella melitensis and BioWars, but we are found Brucella abortus biotype 1 and goat Brucella abortus cases we found it. You are seeing this table, percentage of the abortus and melitensis, like this. And as I mentioned, Brucella abortus biotype 3, 74.6%, and the most common predominant strains in the Turkey for 2021. For this figure, distribution of 338 Brucella strains isolated from cattle in Turkey, the red color belongs to Brucella abortus biotype 3. You are seeing that the east of the Turkey, especially the Erzurum and close cities, most common predominant strains are Brucella abortus biotype 3. Distribution of 53 Brucella strains isolated from sheep in the Turkey. This figure shows that. We isolated from vaccine strains. This is the important things from the sheep. And for goats, 10 goats in the Turkey figure like that. Brucella abortus biotype 1 and 3 colors code is orange and purple, and one strains Rev1 vaccine strains from the Edirne. As a discussion, classical biotype procedure still remains a gold standard performed by reference laboratories to differentiate the classical species and BioWars, according to Scaffolds et al. According to biotype results are almost similar to previous study in Turkey, Erdemli 2009 and 2011, Karagül et al. 2018. These guys work in my laboratory, now they are not. These works belongs to my laboratory. They determined that Brucella BioWar3 is predominant profile in goats. Brucella abortus BioWar3 is predominant in cattle, but Brucella melitensis biotype 3 was dominant in sheep, while atypical Brucella melitensis is predominant now from the abortion agent in our study. As a conclusion, it's important to determine whether the source of abortion is vaccine or infection with the biotyping methods. In terms of disease control, monitoring of the agent with biotyping and some advanced molecular-based methods, such as MLVA, MLST, and VGS, and detection of the dominant biotype and genotype are important for education strategies. Thank you for attention.
Evet. Hocam bize sunumunuz için teşekkür ediyoruz.
Ben teşekkür ederim.
Bir sonraki sunum için Sedat Çam hocamı davet
Yes,
ediyorum.
I am here hocam.
Kapatmam gerekiyor mu? Paylaşımda dur.
Does everyone see the screen?
Evet hocam, görüyoruz.
Okay. Hi everybody, I am Sedat Cham, joining from Harran University. The title of my study is Biofilm Formation of Azotobacter Isolates under Different Temperatures and Phosphate Levels. Azotobacter species are free-living nitrogen-fixing bacteria. They fix free atmospheric nitrogen into the form the plants can use. So it has been used as biofertilizers to improve the productivity of non-leguminous crops, such as wheat, corn, maize, and rice, because of their various plant growth promoting traits. In addition to nitrogen fixation, Azotobacter has other promoting traits, such as production of phytohormones, antagonistic activity, production of siderophones, such as it goes on. This picture shows the nodule formation by rhizobium. Unlike this bacteria, Azotobacter species lives around the rhizosphere, then fix the nitrogen, then plants can use. The use of plant growth promoting rhizobacteria is called PGPRs, has been used as bioinoculant because it offers a promising alternative to chemical fertilizers. As we know very well, chemicals have some detrimental negative effects to human health, environments, underground water, and it's also expensive compared to PGPRs. Many PGPR bacteria can form biofilms, not only on abiotic surfaces, but also on biotic surfaces. This figure shows the biofilm formation by Azotobacter virulence on plants. Biofilm formations provides a number of benefits to plants, such as facilitating resource acquisition, modulating phytohormone levels, and it also reduces the biotic and abiotic plant stress factors. Salinity stress is one of the most important stress factors, and biofilm inoculation to the roots decrease the salt effect to plants. Here's the experimental methods. We isolated 18 Azotobacter species from wheat rhizosphere. Then morphological and biochemical identification were done. For molecular characterization, DNA extracted and PCR were done based on 16S RNA gene, sequence analysis, and phylogenetic tree. Biofilm assays were conducted under different temperature and phosphate levels and measured by crystal violet staining method. Then statistical analysis were made. As a result of maximum likelihood, maximum parsimony, and Bayesian analysis, we identified 7 Azotobacter coracoccum strains and 6 Azotobacter beijeringicae, 3 Azotobacter tropicalis, 2 Azotobacter virulence. The numbers on the notes shows the results of these 3 phylogenetic analyses with 100 bootstraps. These numbers shows the bootstrap values respectively. Here's the accession numbers of the strains uploaded to NCBI BLAST. This shows the biofilm forming ability of Azotobacter strains isolated from wheat rhizosphere. And Azotobacter isolates produce different amounts of biofilms depending on the strains rather than species. The highest biofilm formation was observed in 6 Azotobacter coracoccum and 2 Azotobacter virulence at 28 Celsius. These 8 strains were selected for further experiment for phosphate and temperature effect. Similarly, our results are in a good agreement with this study. In that study, they found different Azotobacter isolates can form different levels of biofilm production. Here's the results of temperature experiments. The first 6 strains is Azotobacter corococcum. The last 2 is Azotobacter virulence. We tested temperature at 3 temperatures, 18, 28, and 37. In this experiment, the biofilm production of these 8 strains was found to be significant at 28 Celsius, as you see on the figure, compared to either 18 and 37 Celsius. The increase in biofilm production at lower temperatures at 28 Celsius is because of the change in viscosity and also increased production of exopolysaccharides at lower temperatures. As we know, exopolysaccharides are the main component of biofilm architecture. In this study, macrobial exopolysaccharides found to be more stable at lower temperatures. So it favors the production of denser and more robust biofilms. It also increased the amount of biofilms. This is the result of phosphate experiments. We tested these 8 strains at different phosphate concentrations from 0, 10, 50, 100, 200 macromolars. As you see, after the analysis, biofilm formation by these strains significantly increased at lower phosphate concentration. In other words, increase in inorganic phosphate concentration decreased the amount of biofilms. The increase in biofilm production is most likely the increased arginate production under low phosphate condition in this study, because in another study, arginate production by Azotobacter virulence increased at lower phosphate concentration. There is a link between arginate and phosphate. As I said before, exopolysaccharides are very important, and arginate is an exopolysaccharide which plays an important role in forming the 3-dimensional structure of biofilm. Under phosphate limitation, enhanced increased biofilm production in Azotobacter isolates is most probably regulated by GICS, GICA2 component signal transduction system. Because in another studies, this regulatory signal system regulates control arginate production in Azotobacter virulence. In addition, another 2-component system regulates biofilm formation in response to extracellular phosphate concentration in Acrobacterium tumefaciens and Pseudomonas aureus. But no study explains 2-component signal transduction system regulates biofilm formation in Azotobacter species. So we concluded this. The biofilm formation is important because in the past, inoculation of planktonic cultures to plants' roots was carried out. But recently, most studies have focused on biofilm inoculation to the plant roots because it has more benefits. Here's a reference and any questions. Thank you for listening to me. I can answer the questions.
Evet, Damla Hocam.
Thank you for your presentation. It was really detailed and very heavy work for presenting. And if there's any question at the end of the session, you can reply them. Thank you. Buyurun Bekir Hocam.
Evet, 13. sunuma atlıyoruz, 14. sunuma geçiyoruz. Dear İman. Doğru mu? Evet, burada.
Merhaba.
Merhabalar.
My name is Iman Abdulkader. Just 1 minute, please.
No problem.
İman hocam önce slaytı açıp küçültün, daha sonra aşağıda ekran paylaşımı var.
I speak English, please.
Okay, so first you need to open your presentation and then there is a green button, share screen.
I don't know what happened. Just 1 minute, please.
Okay, we are waiting, don't worry. When you say in Turkish the greetings, that's why I spoke Turkish. I thought that you have now the Turkish.
No, because I know a little Turkish.
Okay.
Girişi güzeldi, evet.
Your entrance was so beautiful and so native. That's why I thought it.
Türkçe biliyor zannettik.
If you have any technical problem, we can pass the other or then we can come to you. You choose it.
Okay, go to Benkin Hasan, please.
Okay.
Tamam. Hasan'a geçelim o zaman.
Evet hocam.
Evet, dear Hasan. Hasan burada mı?
Hello, how are you, everybody?
Hello.
Do you hear me, please?
Now we hear you. Before we didn't.
Okay, I am Bengina Trushi, the student in master degree in Haran University. I would like to present my presentations, so I will just put it in the screen. You can see it now?
It is visible. And can you try for changing the pages?
Now it's full screen.
No.
Again, I will share it, try to share it again.
Okay.
So,
Bu full screen'de sıkıntı oluyor sanırım Damla Hocam.
Hocam o bilgisayardan ve Zoom'un kendinden kaynaklı diye, yani çoğu kişi full screen açabiliyor ama kimisi böyle bir teknik problem yaşıyor. Now we see your introduction section. If you wish, you can pass the pages by manual with your hand.
okay.
Good luck.
Thank you very, very much. It's my presentation for the biochemical and molecular characterization of Klebsiella pneumoniae isolated from urine tract infection UTIs. So, introduction. You can't see my. Klebsiella pneumoniae is a gram-negative bacterium found in the human intestine, mouth, and skin without causing harmful infection to the human physiology. In the spirit of to another region to the body, it can cause a number of diseases, including urinary tract infections, UTIs, especially in the hospital. UTIs is the most frequent bacteria infections, and the Klebsiella pneumoniae was one of the most common pathogens responsible for UTI. Since the Klebsiella pneumoniae shows the resistance of the most antibiotic currently used, despite of antimicrobial therapy, infection has significant fatality rate in Klebsiella pneumoniae, has immigrated that serious global health concerning and characterization of the diseases has vital importance. Therefore, the isolation of and characterization of the Klebsiella pneumoniae in the samples taken from the patient with the UTIs was aimed on this study. In total of 170 samples are collected from 2 hospitals on the hook from the patient, male and female, of age range between 10 to 70 years. The period between 1st of March to the 10th of October 2021. The samples were analyzed microscopically and biochemically and molecularly. 27 out of 170 samples were identified to be positive for Klebsiella pneumoniae. The multi-drug, then when the result is, can you see my presentation, please? Hello? Do you hear me? Hello? Hello? Hello?
Yes, we see right now. Just change the slides.
Now the slides has been changed. Do you see it?
Yes, we see it. No problem. Go ahead.
Okay. Then this disk diffusion method was used to analyze the sensibilities of 25 isolated of 15 most common used antibiotics. While all the isolated were found to be resistant and to the cerdipidine and simvastatin, the rate of the amoxicillin and cholinergic acids and penicillin rifamycin resistance was 97%. Against the antibiotic of piperacillin and ciprofloxacin rate was determined as 95% to 90 respectively. The multi-drug resistance bacteria like Klebsiella pneumoniae are one of the main challenge of the clinical applications. It is a characterization has vital importance in terms to develop a proper treatment. The applications of the appropriate treatment will be improve the patient quality of the life while also lowering the hospital cost burden. Experimental methods. Experimental method, first we have the collections as a clinical samples as transferred by the sterilized tube. Then we did the general reading of the urine under the microscope. After that, we do the urine culture by Makongi agar, which appears after incubations at 37 degrees for over 24 hours, shows the pink colonies because they fermenting the lactose. Then we did the diagnosis by the Ventec, which is a fully automatic system to identify the sensibilities of the antibiotics. Then we did the bacterial isolation, extraction of DNA for the Klebsiella isolate according to Taha Yassin recommendations. Then we did the PCR as to identify the Klebsiella. Then we had the results. This is the confirmations, the figure for the confirmation of the Klebsiella isolated done by the real-time PCR in our lab. The antibiotic which concentrated to be used in this study, this is the 15 common antibiotics commonly used and currently, which is important to identify resistance and sensibility for the Klebsiella pneumoniae. This is the result and discussions. This is a chart shows the percentage of the 15 antibiotics for the Klebsiella pneumoniae. This is exactly the experimental results, which is we have done for the 25 samples for the 15 of antibiotics such as amoxicillin, and penicillin, and such are all down to the tetracycline. The antibiotic resistance of the Klebsiella pneumoniae isolated, sensibility of 25 isolated of Klebsiella pneumoniae was examined toward 15 different antibiotic use disk diffusion methods as a beginning. Then we have used the Vitek to accelerate the result. which is recommended by National Committee of the Clinical Laboratory Standards, NCCLS guideline. The results show that Klebsiella isolated were resistant to the antibiotic cephalosporin group, and including the cephadrine, cephalothioline, and cefoxamine, and ceftriaxone. All the isolated 100% were resistant to cefaradine and cephalothioline, while 90.5% and 87.5% of the isolated were resistant and cefoxamine and cefoxamine, respectively. The results agreed within Amin 2009 Pakistan, who is noticed that the percentage resistance of cefoxamine and cefoxamine were 82% and 85% respectively. However, reported that Klebsiella were 25 resistance on the maybe related to the process of B-lactamase enzymes, which are able to activate cephalosporin through the cleavage B-lactam ring of the drug. So, and the end of the calculations, the main of the reasons serious problem due the significant increase of the resistance of the bacteria to antibiotic, broad spectrum antibiotic due to the excessive use of the antibiotics. Resistance of the bacteria that cause urinary tract infection pose a great danger to the public health because of the resistance to a different group of the antibiotic. The ability of the bacterium to process the biofilm of the most important factors on the virulence of the bacteria, what is confers the bacteria that resistant to the most antibiotics. Thank you for the attention. I hope I delivered most of the information. If any questions, I will leave my email address. And sorry for hesitation.
No, it was really perfect. Thank you for your presentation. We really appreciate.
I appreciate also your help and support. And thank you very much. I will leave my email address for any questions.
Okay. Thank you.
Evet, Aylin Hocam'dan devam edelim. Aylin
Evet,
Gökmen.
merhaba. Buradayım.
Merhabalar.
Hemen ekran paylaşıyorum.
Tamam Aylin Hocam.
Görünüyor mu acaba?
Evet, görebiliyoruz hocam.
Tam ekran görüyoruz. Bir sayfayı değiştirin olmadı hocam.
Tamam.
Mükemmel. Teşekkür ederiz. İyi gidiyor.
Tamam, ben teşekkür ederim. Good evening, dear participants. First of all, I would like to thank Congress organizing team for supporting us and for your participation. Sorry. I'm Aylin Gökhan. I work as a specialist medical doctor in the Department of Histology and Embryology at Ege University Faculty of Medicine. Today, I will give you information about my specializing thesis, which was about the histological investigation of the rat ovarian tissues that we vitrified with PRP supplemented vitrification solution. I have planned to present our work in the following order. After a short introduction, I will give you information about the method of the experiment, and then I will share the results we obtained, and I will summarize the results and results achieved with the experiment. Various substances have been tried and positive results have been obtained with vitrification. I'm so sorry, there's a problem in my computer. I will do something, sorry for that.
Now it is in the size.
Because Zoom comes my thing.
But we are seeing, don't worry. You can follow the presentation.
Okay. I will do it again. Is it okay?
It is.
Okay, I will continue. Various substances have been tried and positive results have been obtained in reducing cellular damage caused by cryopreservation and cryoprotective agents. Platelet-rich plasma has been the subject of our study to reduce cryo damage. PRP is a blood product obtained by condensing platelets. It is rich in growth factors. It makes a great contribution to the natural healing process. So it has found a lot of use in regenerative medicine. We aim to improve our ovarian tissue cryopreservation methods, considering that the addition of PRP to the vitrification solution, and this will contribute positively to tissue viability after warming due to the various growth factors it contains. 8 to 12 weeks old, 21 female viscerals were used in this study. Experimental animals were randomly distributed to the following groups with 7 animals in each group. Group 1, control, fresh group, no application was administered to the rats. Group 2, the vitrification group in which the vitrification solution was supplemented with PRP. Group 3, PRP negative group, the vitrification group in which the vitrification solution was supplemented with PBS. It is PRP free. Leftovers in the rats in group 1 were transported to the laboratory in fixative solution for histochemical, immunohistochemical, and inflorescent stains. The right and leftovers of the rats in group 2 and group 3, which were transported to the laboratory in the base medium, were vitrified and stored in liquid nitrogen at minus 196 degrees Celsius for 1 week. In order to prepare PRP from the rats in group 2, 6 milliliters of blood was harvested via cardiac puncture and the ovaries were removed from all rats. Right ovaries of rats in all 3 groups were transferred to the biochemistry laboratory under cold chain condition for myeloperoxidase, malondialdehyde, and catalase analysis. We adopted chance method for PRP preparation. After cardiac puncture, the blood was anticoagulated with sodium citrate. The blood samples were centrifuged at 2 spin. You can see here. And after first spin and second spin, lower part was activated with calcium chloride, and it was made ready for use. PBS was used instead of PRP in group 3. He et al. Model was modified for the vitrification process. Tissues were kept in equilibration solution for 15 minutes. Then they were kept in vitrification solution 1 for 2 minutes. And directly taken into a storage tank containing liquid nitrogen. In group 3, the process proceeded in the same way, only the vitrification solution did not contain PRP. Tissues were stored for 1 week. After 1 week, tissues were removed from the tank and treated with the warming solution 1, 2, and 3, respectively. Then the tissues were washed and incubated in the incubation solution. Then histological follow-up was started. Tissues were fixed with 4% formaldehyde. Routine laboratory procedures were applied. Tissues were embedded in paraffin and blocked. Hematoxin eosin, periodic acid sheaf, and malaritrichrome were applied to the sections obtained from the blocks. In addition, as immunohistochemical staining, connexin 43, box, BCL2, caspase 3, tunnel assay was performed. Tunnel and caspase 3 immunofluorescence staining were performed as immunofluorescence staining. And we also performed biochemical analysis. According to the biochemical results, MDA and CAT elevation in group 2 and CAT elevation in group 3 were significant. Group 2 was the most damaged group according to the results of histochemical studies. The results of the immunohistochemical studies also confirmed that group 2 was the most damaged tissue. In caspase 3 and tunnel immunofluorescence examinations, immunopositivity was higher in group 2 than in group 3. As determined by statistical results, the group containing PRP in the vitrification solution was more damaged than the group PBS contained. Immunopositivity in caspase-3 immunofluorescent and tunnel immunofluorescent in group 2 was statistically significant. In order to develop ovarian tissue cryopreservation techniques, first time in the literature, the whole rat ovarian tissue was vitrified with a vitrification solution supplemented with calcium chloride activated autologous PRP. Primordial follicles were largely preserved in experimental groups. The degeneration of primary and secondary follicles was more common in the experimental group, especially in group 2. Other degenerative findings were also more remarkable in the group in which PRP was used. For the first time in the literature, whole rat ovarian tissues were vitrified by adding calcium chloride activated autologous PRP to vitrification solution. In rat ovarian tissue vitrification using EG and DMSO, primordial follicles were preserved. Significant damage occurred in follicles in cortical and medullary areas. If ovarian tissue is planned to be cryopreserved to restore ovarian functions after recovery, and to supply oocytes that can be used in IVF procedures in cases with the possibility of developing POE due to the cancer or non-cancer reasons, it has been concluded that preserving the ovaries as small cortical pieces that the follicles are located in, rather than as a whole, may yield more successful results. To increase the success of the vitrification method and to ensure standardization, it is necessary to determine the appropriate cryoprotective agents and to test them in human and animal studies with different combinations and different densities. We use PRP, which has a low probability of triggering immunity due to its autologous nature and has a high probability of supporting cellular viability instead of PBS in our study, aiming to reduce cryo damage and CPA toxicity and not to use animal-derived additives. However, in the light of the findings we obtained, it was concluded that calcium chloride used in PRP activation caused more cellular damage. To observe its protective effect in rat ovarian tissue vitrification, the PRP to be added to the solutions should be activated with a lower amount of calcium. Moreover, the application of PRP activation with different activation methods or non-activated PRP should be considered. Testing PRP with slow freezing protocols in OTCP, ovarian tissue cryopreservation, will be beneficial in terms of developing cryopreservation protocols. Transplantation of soft ovarian tissues after cryopreservation and observation of ovarian functions will provide important data. Thank you for your attention. And I will give my email address. You can ask me with email.
Evet hocam, teşekkür ederiz sunumunuz için.
Ben teşekkür ederim. Kusura bakmayın. Zoom'a alışık değilim. Okuyacağım yani metinlerin üzerinde hep şey vardı, Zoom vardı. Onu çekmeye çalıştım. O yüzden biraz
Biz
zor
hiç
oldu.
dikkat etmedik bile, yani görmedik bile onları, sıkıntı yok. Tekrardan teşekkür ederiz. Next presentation, İman.
Okay.
Can you share your presentation, Iman?
Yes.
Could you solve the problem?
Can you see it?
No, we cannot now.
Okay.
I think she has also internet problem because her screen has stuck.
Sanırım Damla Hocam dondu mu?
Evet hocam, internetinde de bir problem var. Ekran dondu. Değişmiyor. İsterseniz biraz bekleyelim.
Tamam, olur.
Ya da sorular varsa İman bağlandı. Şimdi başka bir bilgisayardan bağlandı galiba. 2 tane İman görünüyor Zoom'da. And also, if there is any question, we can receive them.
Me now?
We are seeing now.
Okay. And my presentation also?
I'm seeing myself. Yes.
Okay. It's good now?
It's good. Can you change the pages for trying?
Okay. Now?
Perfect.
Evet.
Okay.
Good luck. Bye-bye.
My name is Iman Abdelkader. My presentation for characterization of Escherichia coli from the isolates of the born wounds. Bacteria with resistant to the currently used antibiotic are emerging as a serious global health problem and one of the main issues to be death with unborn patients. Despite significant advance and important treatment, the risk of infection-related death in born patients remains relatively high. Therefore, it's critical to identify and characterize the bacteria that cause infection in born patients. Born wounds are commonly associated with Escherichia coli. Therefore, the characterization of Escherichia coli in born wounds is critical due to having the capacity to cause different complications and aim to investigate in the study. Clinical samples were collected from two hospitals, Azadi Hospital and Hoogborn Hospital in The Hague, and I take it from these patients in the period between 1/3/21 to 10/10/21. The Vitic2 system was used to explore the identification of bacterial profiles in the samples as well as their suspectability. The genotyping of Escherichia coli in the isolates was determined using the PCR technique after performing DNA isolation. 25 samples out of the 147 samples were identified to be positive for Escherichia coli. All of the Escherichia coli positive samples were found to be resistant to cephalothin, cephradine, piperacillin, and rifampin, while the rate for amoxicillin plus clavulanic acid and ampicillin was 9600%. This rate was found to be 9200% for amikacin and cefotaxime. The multidrug resistant bacteria like Escherichia coli is one of the main challenges for clinical applications, and its characterization has vital importance in terms of the development of a proper treatment strategy. The development of effective medicine will improve the patient quality of life while lowering the mortality rate. Our research is beneficial, and as to the body of knowledge, it would be beneficial to replicate the study with bigger populations. Experimental methods. First, I collect samples from the hospital, and then I take a swab from the patient, and then I prepare cultures, Ixmacondia agar, and then diagnosis by Vitic2. Bacteria isolates, extraction of DNA from the E. coli isolates, PCR assay, and then results. This line, just 1 second, please. Okay. This figure, PCR confirmation of Escherichia coli isolates, line 1 to 100. Kb plus DNA marker, and lines 2 to 25 samples, line 26 positive control, line 27 negative control. This is the antibiotics and construction used in the study. Amikacin, amoxicillin, clavulanic acid, ampicillin, cefotaxime, ceftriaxone, cephalothin. I used 15 different antibiotics, and I have this schedule for the this sample, and this is the chart title of antibiotics resistant of E. coli, and this one. This is the table show the result of resistant the E. coli used in the experimental. As you see, we have used 15 common antibiotics tested in our samples, identified it as a positive of E. coli. Antibiotics resistant of E. coli, susceptibility of the 25 isolates of E. coli was examined towards 15 different antibiotics using this diffusion method recommended by the National Committee for Clinical Laboratory Standards guideline. Results show that E. coli isolates were resistant to the antibiotics of cephalosporin group, included cephradine, cephalothin, cefotaxime, and ceftriaxone. All isolates, 100%, were resistant to cephradine and cephalothin, while 100% and 8700% of isolates were resistant to cefotaxime and ceftriaxone, respectively. The result agreed with Amin et al. 2009 in Pakistan, who noticed that the percentage of resistant to cefotaxime and ceftriaxone were 82.5% and 8500 respectively. However, Nasihi et al. 2010 reported that E. coli were 2700% resistant to ceftriaxone, and this may be related to posing of B-lactamase enzyme, cephalosporins, which are able to then inactivate cephalosporin through cleavage B-lactam ring of the drug. Finally, conclusion, finding of the study suggests that multi-drug resistant gram-negative organisms are the most common isolates from bone wounds. Hence, a careful selection of antibiotics to treat bone wounds infection is required for proper management to these wounds in order to help reduce morbidity and mortality associated with multi-resistant bacteria. Thank you for your attention.
We thank you. Your effort for solving the technical problem, but the presentation was perfect. Thank you very much for your work.
Thank you. Teşekkürler, çok teşekkürler.
When you say this, we understood that you know Turkish.
Yes, ben biliyorum, but ama biraz.
Okay, that's enough for us, but your English was so fluent. Thank you. Thank you for the presentation. Buyurun Bekir Hocam.
Evet hocam, sanırım sunumlarımız bitti.
Evet.
Evet. Bugünkü oturum güzeldi, başarılı bir şekilde götürdük. Ben
Çok teşekkür ederiz destekleriniz için. Çok keyifli bir oturumdu.
Aynen, ben de öyle katılıyorum. Umarım sunumu yapan arkadaşlar da, hocalarımız da aynı fikirdedir.
Evet, çok güzeldi. Teşekkür ederiz.
Biz teşekkür ederiz. Dear researchers, thank you very much for your attention and for their presentations. It was a so friendly and academic platform for sharing the researches and knowledge here. And if there is any question, most of the researchers share their email addresses, and I cannot see any question now. If there is any question, you can directly ask now. We can wait. Or if you wish, you can directly ask to the corresponding author. Sayın hocalarım, hocalarımızın çoğu e-mail adreslerini buraya ifade etti, belirttiler. İsteyenler paylaşabilir. Sorularınızı şimdi de alabiliriz. Dileyenler direkt sorumlu yazarlarımızla iletişime de geçebilirler. Sizlerin görüşlerini bekliyoruz. Ya da söz almak, katkıda bulunmak isteyen varsa söz verebiliriz. Sizin için de uygunsa herhangi bir görüş gelmedi. If it is suitable for you, we will follow our schedule for tomorrow. So see you tomorrow. Herhangi bir katkı ya da soru yok şu an için. Yarın görüşmek üzere. Kendinize iyi bakın. İyi akşamlar.
Bye bye. İyi akşamlar.
See you tomorrow. Have a nice day.
Thank you.
Bekir Hocam çok teşekkürler katkınız için.
Ne demek Damla Hocam. Ulaş Hocama da selamlar. Görüşmek
Getirim
üzere.
Hocam çok teşekkürler. We really thank you for Professor Dr. Bekir's help and support for the Congress. See you tomorrow. İyi
Teşekkürler.
akşamlar hocam. Görüşmek üzere.
Görüşmek üzere tekrar.
See you everyone. I'm closing the session. See you tomorrow.